All right, well, thank you all for coming. Good turnout on a beautiful day. Today we're going to talk about clinical trials and its role in drug discovery and development. And we have Rich here, Rich Whitley, who is, I think, a little bit of a UAB celebrity, considering the turnout today. And just so you, I sent an email around this morning, but the speaker for next week ended up having a conflict. Actually, the conflict is that they are changing jobs. And they didn't really know that until recently. So unfortunately, we cannot have that seminar next week, but you can go to the website and look at the slides from last year if you really want to know about toxicology. But then the week after that, we have Mark Rivera. He's the Director of Business Development at Southern Research. He comes and gives a presentation, and he explains really nicely how you make connection with the company to maybe form a partnership if you brought a drug discovery project to a point of where a company might be interested in it and how that process works. So I think a lot of us are interested in the business side as well. But today we're going to talk about clinical trials. So Rich, the floor is yours.
Great, thank you.
I'm going to close this door.
Yeah, good. I want to keep this as informal as possible. So don't hesitate to stop me, or if you have questions, raise your hand, jump up, shout, scream, whatever you want to do. For me, this is one of the really fun aspects about drug discovery and drug development, because ultimately it's like a siphon. You've got to come down to one final point, and that is you've either got something or you don't have anything. So what I want to do today is just review with you the kinds of things that we do in clinical trials, but with a focus on those that lead to approval of a new drug application by the Food and Drug Administration.
I do apologize. I've got to get this so that this is right.
You do what you
Oh,
need to
okay.
do.
Technical
Good.
support. Thank you, Brian. So that's probably why it didn't work last 2 weeks.
So the first aspect you want to remember is any clinical trial is predicated on the understanding of the natural history of disease. You've got to be able to define your endpoints, because the endpoints are going to be the basis for any protocol that you perform. And that's going to be the basis of anything that the FDA reviews in terms of licensure. And the example that I'm going to use again and again is the impact of congenital cytomegalovirus on hearing. And I'll show you some of the kinds of things that we've learned over the years. But don't forget, we don't always talk about drugs when we look at licensure by the Food and Drug Administration. We can talk about evaluation of a diagnostic assessment, or we can talk about the assessment of an intervention. For example, placing a stent for individuals who have coronary artery disease. But when you think of drug discovery, and for the most part, when most people think of drug discovery, it's the appraisal of a drug. Now, there are three forms that can take. One is it's a placebo-controlled study, and that's when there's no defined therapeutic intervention for a given disease. And there are only a couple exceptions to that. The one exception that I can think of is rabies, where we know that if you develop rabies, it's uniformly fatal. And we've had that discussion with the Food and Drug Administration. And if you have a therapeutic that you think works and want to try it experimentally, they'll probably let you do an uncontrolled clinical trial that could lead to licensure with enough individuals entered into the study. You can do a drug-drug comparison, and you'll see that all the time. And we're going to come back and talk about the specifics of that in a minute. And then you can do open-labeled evaluations. And open-labeled evaluations usually are in the form of phase 4 studies. So as we think about where we can go, and let's see if we can get this to move forward. And it's frozen.
Oh, probably now. There we go. It should work now.
Okay, so first of all, you have to ask the question when you're assessing a clinical trial, who is the sponsor? And the goal is to identify bias. So if industry is the sponsor, remember, they want to do it for one reason, and that is to make money. And that's not to say that it's bad, it's just to say that there's an inherent bias when industry is doing a controlled trial. And I'll come back to that again and again and again, because it's part of the issue of what we have to deal with here at UAB as an academic institution. Now, the sponsor can be an academic institution, and obviously the goal here is to advance knowledge. Investigators want to publish, and publish. But there's added value as well, and that is to make money if the institution, in fact, has a vested interest in the development of an intervention. And I'll come back and talk about that in more detail. But clearly the institution would like to establish proof of principle. And proof of principle means that if the institution owns the patent or owns the drug, and you have proof of principle data, at the end of the day, you'll be able to translate into a higher monetary reward. And then the third sponsor is an individual. And the goal of the individual is to establish proof of principle and the value thereof, and also to advance a career. So what are the components of a clinical trial? The first is, what is the hypothesis? Any clinical trial, I don't care which one it is, even if it's for licensure, has to be grounded in a hypothesis. And that hypothesis can be drug A is better than drug B, and then you have to specify what you think that difference between drug A and drug B is going to be. For example, drug A will decrease the mortality of a disease from 50% to 20%. And the reason that becomes important is because that becomes the standard that not only the investigator, but the Data Safety and Monitoring Board and the FDA will use when they evaluate the results of a clinical trial. But it's also the basis by which you're going to calculate your sample size. And that will determine exactly how you proceed. So that if the difference is 30%, the number of individuals required for an adequate sample to answer the question is going to be far less than if you want to decrease the mortality from 50% to 45%. So we'll come back and talk about that in a minute. So it's grounded in a literature search, and we talked about outcome A versus B. So non-inferiority is something you always need to consider. And if you pick up the literature, and you can go to NEJM and you can look at the last month, and there have been 2 AIDS clinical trials that have been published in NEJM, each one having about 4,500 individuals in it. They were non-inferiority studies. So the difference between the 2 treatments, and one is the treatment of choice, and the other is the new treatment, can be no more than 10%. Now, there are some statisticians who say it shouldn't be any more than 5%. But if you go to a 5% variance in outcome between the 2 treatment groups, showing non-inferiority, that sample size goes up to 20,000, 30,000. And that just doesn't become feasible for any institution, any pharmaceutical company, or any individual to do, unless it's population-based development. And then the last question is it ethical? And you have to always remind yourself, am I doing the correct thing by my patients? Am I doing that which is going to really help them? So that will translate to endpoints, and you'll have primary endpoints, secondary endpoints, toxicity endpoints, and certainly exploratory endpoints. Don't diminish the value of exploratory endpoints, because exploratory endpoints will teach you where the next study needs to go. So for example, if you're using PCR as a tool that will be a diagnostic in a study as opposed to isolation of virus and cell culture, then you want to prove the value of PCR as well. So what are the first steps? You've got to develop a management plan, and that's where it begins. Who will manage the data, and where will the data be housed? That is fundamental to any clinical protocol. You're talking about human subjects. So point 3 here, how is it protected? You've got to protect the confidentiality of an individual's rights. So the data have to be locked, and they have to be secure. When you write a manuscript regarding a clinical trial, one of the things that the editor will ask you is, did you secure the data? How was it secured? Tell me more. And then how are the data verified? So we do double entries, we do audits in the field to make sure the actual data match up with a patient's file. So, I mean, that sounds trivial, right? But it isn't. If you look at what happens in some of the Eastern European countries, one case record form looks like another case record form, looks like the third case record form. So you can't back it up by any patient file. All somebody did was say, oh, this looks like the right outcome for this patient, and the data are totally false. So you've got to make sure it's real. Then you have to have an adequate biostatistical design. Again, that's not trivial. Sample size, how's the randomization going to be done? And then you have to have a data safety and monitoring board for any clinical trial that's being performed. It's de rigueur today. The very first clinical trial that was used at the NIH goes all the way back to the 1970s, and it was actually a study that was done here on herpes simplex encephalitis, where outcome for placebo recipients was compared with treated recipients, And what was learned was the mortality was decreased from 70% to 28%. That caused the Data Safety and Monitoring Board to say, terminate the placebo control. And then you have to have an analysis plan. The analysis plan is essential for the FDA.
A question about a DSMB. Mm-hmm. If you have a multiple site study, will each site have its own DSMB, or is there one for a whole?
There's one for the whole clinical trial.
Okay.
And hopefully in this country, and I have to say UAB has taken the lead on this, you have a universal IRB so that you only have one institutional review board that will cover all of the other institutions that are participating in the clinical trial. There's always variability with some institutions, and you can't say it's going to be 100% because lawyers always want their stamp on what happens with a clinical trial. And whether I like it or not, I've argued for universal institutional review boards forever. Mm-hmm. Best example being West Nile virus infection, because you can't predict where it's going to be. So if it's going to be at St. Vincent's Hospital, they don't want to transfer a patient to University Hospital just because our IRB approved it. So we need a universal institutional review board. Okay, so what happens behind the scenes? There's got to be a statistical review, and that requires buy-in from the Food and Drug Administration. Even if I'm doing a study that's not a licensure study, it's got to be filed with the FDA, and the FDA has to agree with my analysis plan. That's not to say that they're always willing to roll over and play dead and agree with you, because they don't. I can assure you of that. There have been a lot of knockdown, drag-out battles with them about what the control should be and what the power of the study should be. But it's a good dialogue, and it's a healthy dialogue. You have to design case record forms. They have to be uniform. And then you need a series of approvals, and we can't trivialize the approvals. One is an institutional review board, and fortunately UAB has a terrific IRB, and it works between UAB and Southern Research as well. We also use the Western Review Board. And then really important that we need to always remember is that we have to declare our conflicts of interest. So we have a conflict of interest review board at UAB, and that's unbelievably important. For example, if I were to do a drug study for Company X, and I own 100,000 shares of Company X's stock, and I'm studying their drug, it's to my benefit if the outcome is in favor of Company X's drug. That's a bias that's got to be declared. I can't do that clinical trial because I have a conflict of interest that is greater than would be allowed by the institution. So we have to be real careful. And in large part, it's judgment. In large part, it's the ethics of the individual. But one must always be careful that you declare that conflict of interest. And just so you know, what happens is every year investigators at this institution have to file a management plan with our conflict of interest review board. So for example, if I'm on a data safety and monitoring board for the NIH, my conflict of interest review board here needs to know that. If I'm on a data safety and monitoring board for GlaxoSmithKline or Merck, they similarly need to know that, even though I don't receive compensation for it. It's essential for our conflict of interest review board to know that. And then if I'm doing gene therapy, which we've done with a virus that we made here that's now in Jim Markard's hands for patients who have glioblastoma multiforme, that introduces another variable, and that's gene therapy. And so we need to have our own gene therapy review committee take a look at it. Sometimes that'll be punted to the recombinant DNA advisory committee at the NIH. Right now, I chair the recombinant DNA advisory committee, and it's my goal to make sure it's disbanded by the end of this calendar year if I can do it, because it serves no legitimate purpose. But you read about it in the newspaper. For example, the first CRISPR-Cas protocol that's been put into place was approved for Carl Jung's study at the University of Pennsylvania. That needed RAC approval. If we were to do a CRISPR-Cas protocol here or a CAR T-cell therapy here now, it's been reviewed enough times at the recombinant DNA advisory committee that it would only have to be seen by the gene therapy review committee. I mentioned briefly about universal IRBs. You'll see that happen more and more often. That's particularly true with oncology protocols. And they've been leading the way there through the National Cancer Institute. Then the last thing that happens behind the scenes is you have to have an investigator's brochure. And I'll tell you a little bit more about that, but the protocol can't go to the FDA without an investigator's brochure. So what's the protocol? Background and significance, why you're doing the study, approaches to the problem, and here one has to be real clear to declare any competing protocols that are being performed in the community. So that if there's the chance that someone's going to improve therapy for the disease that you're studying, you need to tell your data safety and monitoring board that, because it will change the ethics of the study that you're performing. You have a background section which will include relevant data, and that will be cell and animal experience as relevant. If it's a drug, its absorption, distribution, metabolism, excretion, and potential toxicity. And what was learned in terms of the maximum tolerated dose for toxicity. Endpoints, inclusion, exclusion criteria, details of evaluation. And what that means is you have a flow sheet. And the flow sheet is a huge sheet of paper that has every single thing you're going to do to that patient during the study. And what we tend to do is, we do it. We give each patient who participates in a clinical trial that flow sheet so they know exactly what's going to happen to them and why. You need to then make sure in the protocol itself the sample size is determined, including the statistical methodology, how adverse events are not only going to be monitored but be reported. So if it's a serious adverse event or a life-threatening adverse event, we report them within 24 hours. And each site must report them to us if we're doing a multi-center study within 24 hours. That's got to be declared to the FDA immediately. Adverse events can be reported in a more relevant fashion. They don't have to be immediately reported. The literature that's cited, then you have appendices that'll include case record forms and the investigator's brochure. And the protocol obviously is filed with the FDA. Now, what about the Institutional Review Board? We need to remember a couple fundamental things. There's a difference between informed consent versus assent. You know, if we're talking about a minor, assent is required as best we can determine that. Obviously, parents like to speak for their offspring. And if you've got a 2-year-old who's got acute lymphoblastic leukemia, it's hard for that child to know what the word assent, quote unquote, means. But the parent has been explained exactly what happens and can make a judgment accordingly. The language of the informed consent has to be understandable to a teenager. We tend to overwrite informed consents, and that tends to be a problem, but that's picked up immediately by our own institutional review board. It's got to be accurate and without bias. And as we increasingly learn, as we recruit patients from different backgrounds and different heritages, it has to be in the language of the volunteer. We have to evaluate the mechanism by which consent will be obtained. There's got to be equipoise. There can't be conflict of interest. Informed consent should be obtained by the principal investigator. A lot of times it's obtained by a surrogate who is a research coordinator after it has been explained to the volunteer by the principal investigator. And don't forget, we have marginalized populations, and that's children, women, unrepresented minorities. And if you lived in Galveston, Texas, it's the penal system of the state of Texas, where they frequently recruit volunteers from the prison system. They're marginalized, and you have to remember that, and it requires special review by institutional review boards. What about conflict of interest? The goal is to ensure that all parties have no vested interest in the outcome of the study. And it could be financial, or it could be non-monetary gifts in kind. And the example would be, well, I'm going to give you a ticket to the Alabama-Auburn football game. Can't accept it. Now we have CMS rules whereby the pharmaceutical industry declares every penny that they spend on physicians, whether it's taking you out for breakfast for $2.50 or taking you out to lunch for $5. But there are real issues that will get reported. And for some people, those issues are greater than others. And we'll see here, even at UAB, that there are investigators who have done studies whereby they have received significant monetary payments. And it tends to end up in the local newspaper or in the New York Times or in the Wall Street Journal. And if you just declare it to your institution up front, you can avoid a lot of headaches. Investigators, as well as their family members, have to declare potential conflicts. So you may not own 100,000 shares of stock in the company, but your wife may own 100,000 shares of stock in the company. That's a huge conflict of interest. The people would like to say, oh, that doesn't influence my opinion, but it really does at the end of the day. There's another conflict of interest that we don't talk about very much, and that is what happens when the data come in. Who writes the article up? And the article can be written either by the principal investigator of the study, or it can be written by the pharmaceutical company. If it's written by the pharmaceutical company, that's what we call ghostwriting. And usually it's written to suit the pharmaceutical company's needs, and it may not accurately tell the story of the data itself. If that's the case, certainly this institution doesn't allow such ghostwriting to take place. And then, does the institution have a conflict of interest? And we're seeing this more and more frequently. And the best example I can give you is Jim Markert and I made a virus that we wanted to use to treat glioblastoma multiforme. And in fact, we had the National Cancer Institute produce the virus. We got IRB approval from Vanderbilt because of the conflict of interest with our own IRB to do the clinical trial. And then Jim had to find an independent investigator in the Department of Neurosurgery to do the clinical study. UAB holds the patent. UAB stands to receive monetary compensation. UAB has a conflict of interest, as do the investigators. So it's a point you need to keep in mind and declare. Yeah.
If you, for example, if you're going to do trials here, but get review board at Vanderbilt, do you pay for the review?
It's a quid pro quo. We do the same thing for Vanderbilt.
Okay.
So we're not paying Vanderbilt to do it. But when they have a patent that is going to lead to a clinical trial there, we'll do the same for them here. What about federal approvals? The first question you want to ask yourself is an IND required? Well, no. If performed within the state of origin and it does not cross state lines, an IND is not necessary, but don't do that here. Just absolutely don't bother doing it. It's an academic institution, and if you're developing an intervention, you should go ahead and get an investigator-initiated IND, and all the resources exist within the institution for you to be able to get an IND if you have done your homework, dotted your I's, crossed your T's, and put together the appropriate data. If the purpose of your intervention is new drug application directed, you definitely need an IND. You can't do it without an IND. There's an investigator in Tuscaloosa who thinks he has an intervention for fibromyalgia. And he's been doing a series of studies on his patients in Tuscaloosa with approved medications, but for an unapproved indication. For all intents and purposes, that requires IRB approval. It requires informed consent. He's doing it with neither IRB approval nor human consent, and he wants to take it to an NDA. Well, it's not going to happen. Registration of federally funded clinical trials has to appear in clinicaltrials.gov. And that's good and bad. You know what everybody's doing. But the problem is, for example, if I'm doing a clinical trial in neonatal herpes and I'm going to give a drug for a period of 6 months to babies who had the disease with the outcome being improved neurologic outcome, anybody can go online and see, oh, the group at UAB is doing a clinical trial for neonatal herpes and they're giving a licensed drug for a longer period of time. I'm just going to do it. And they go ahead and do it without any form of approval and without the data being reported as either beneficial, harmful, or toxic. And that's what we really need to worry about. And then you need to remember that whatever we do in clinical trials has to comply with the Code of Federal Regulations. And the Code of Federal Regulations is going to drive you nuts. I mean, it's even to the point where if you have data being entered by a fax machine, you have to verify that the fax machine is reliable and the data are reproducible. As foolish as that sounds, that's important for the Food and Drug Administration. And when you get audited, that's what they're going to look for. So where does funding come from? Ideally, you'd like it to come from the government because that's the most likely non-biased source of funding. And that's the way, at least with our group, we do our clinical trials. We're not doing them for industry. We're doing them with federally funded money for clinical trials that we've written. But other sources of funding can be industry. And that's not to say that it's bad, because investigators at UAB may generate a great deal of income for their division by doing clinical trials, particularly in cardiology and cancer. It can come from venture capital. And venture capital is anxious to see whether or not a new intervention will generate proof of principle data that will allow them to put more money into the targeted development. It can come from a foundation or philanthropy, or it can come from the institution, and that can be the Health Service Foundation here, or it can be a department. Department of Pediatrics puts a lot of money annually into clinical trials, one of which is the study of an engineered virus for glioblastoma multiforme for children that Greg Friedman's doing. So then what are the stages of clinical development? And each stage requires informed consent, IRB approval. If it's a genetically engineered product, it's going to require gene therapy review as well. Phase 0, 1, 2, 3, and 4. So what's phase 0? These are clinical trials to expedite the clinical evaluation of new molecular entities. And it's only been on the books for the FDA for about the last 6 or 7 years. It's really an exploratory IND that performs or allows for the performance of first-in-human testing of new agents at subtherapeutic doses. And the goal really is to make sure that the product can be detected in the blood or detected in the urine, and that there are no untoward adverse events that would have been anticipated. So the kind of requirements are drug target effects, PK and PD. Usually it's done in a very limited number of patients because the Food and Drug Administration wants you to move quickly to either a phase 1A or a phase 1B. What's a phase 1 study? It defines distribution and potential effects of the intervention. Phase 1A is simply pharmacokinetics and toxicity in normal human volunteers, and it entails sequential blood draws and a collection of select biologic fluids. And that could be urine, it could be saliva, it could be cerebral spinal fluid if you're talking about spinal muscular dystrophy or myotonic muscular dystrophy, where the goal is to show that the drug crosses the blood-brain barrier. Phase 1A studies are frequently done in paid volunteers, and they're done at commercial houses that have a stable of basically college students and graduate students who want to get paid to participate in these studies. A phase 1B is pharmacokinetics, pharmacodynamics, and toxicity in patients with a given disease. So for example, if we're doing a 1B study, we could do it in patients who have West Nile virus infection, where we're testing 2 or 3 doses to look at distribution of the medication that's being given. So here's a 1B protocol design, and this is in congenital CMV, and this is a group of babies who have disease. So you give the drug IV for days 1 to 4, and that's shown right here. And then you go ahead and do IV PK after day 4. And then you give oral drug for days 5 and 6, and you repeat the pharmacokinetics. And then you go back to the standard dosing for days 7 to 34. And then when you get to day 35, you repeat PK with oral dosing. You can simplify that protocol because, as you might imagine, the version at the top half of the slide is pretty complicated. Think of what it would be like if you're a parent and you're going to do PK on 5 different occasions. Here with version 2, you're only doing PK on 3 different occasions, and it becomes a much simpler protocol itself. So what do you learn? So if you look at time post-dose on the x-axis versus the concentration of ganciclovir, the drug that was being evaluated, you can basically get an idea of the distribution of the quantity of drug over time and see that it's cleared from the blood. More importantly, what's the difference in the kinetics between day 4 and day 34? Day 4 is in blue. So this is when you give the drug, IV, ganciclovir on day 4. You can see that the peak plasma level approaches 7 milligrams per liter. But on day 34, you only get 4.5 to 5 milligrams per liter of dose. And you can see that the area under the curve is much less with the red line compared to the blue line. Well, why is that the case? Well, what we learned very quickly is if you increase the dose on day 35 and day 36, you can replicate the pharmacokinetics with IV medication. And it's all because of renal function. As a baby's kidney matures, clearance improves, and with improved clearance, the dose of ganciclovir and valganciclovir needs to be adjusted. That's what you learn in a phase 1b study that's done over time. So you know that you're appropriately dosing the patient to treat the disease that the child has. And this is just simply observed versus predicted models of ganciclovir concentrations. Now I want to show you one exploratory endpoint, and this is viral load in the blood as a function of treatment. So treatment's administered for 56 days. You can see that the viral load in the blood is decreased from nearly 70,000 viral copies virtually to undetectable by day 14. And it stays that way until you discontinue therapy. And then toward the end of therapy, drug or virus rather by PCR returns in the blood, indicating that there may be better approaches to this disease than using one drug. And we can talk about that later. What about phase 2 studies? This is really on the path to licensure. You're pretty confident from a 1B study that you've got the right dose, but you want to make sure. It's performed in the target population. It is proof of principle. Usually it's 3 doses of medication, 120 to 160 per cell. And phase 2B, again, is target population. And it's an expanded sample size to guarantee proof of principle. So the FDA likes phase 2 studies. And they very, very rarely will license a drug predicated on a phase 2 study. They've done it, but it has to be a rare disease. A rare disease, neonatal herpes, congenital CMV. And they're important. Now, I want to give you an example of where we can go with a phase 2 study that is proof of principle. Here you have babies with congenital cytomegalovirus infection who had to have central nervous system involvement. Informed consent was obtained from the parents. These are children all less than a month of age. So here you can't get assent from the baby. You can get informed consent from the parents. They're randomized to ganciclovir versus no treatment for 42 days. Why no treatment? Why not placebo? Can't keep an IV in a baby for 42 days when you're talking about a peripheral intravenous catheter or a central line, because the risks thereof outweigh the potential benefits of giving normal saline. Then they're monitored, and clinically, virologically, serologically for toxicity. Then they're followed up serially. Well, what can you learn from a study like this? The primary endpoint was brainstem evoked responses, both biologic assessment, total number of years, functional assessment, best year. Secondary endpoint is laboratory improvement by 2 weeks and clinical improvement. Now here's the outcome. Ganciclovir recipients are shown on the left. This is change in hearing between birth and 6 months. In the no treatment group, the red pie graph is deterioration in hearing. Green is stabilization or improvement. You can see in the ganciclovir recipients, there was stabilization of hearing or improvement, whereas there was deterioration in 40% of the no treatment group. What happens if you follow these patients longer, for greater than a year? You will see some deterioration in hearing in the ganciclovir recipients, but not nearly to the extent that you do in the no treatment group. What does this teach you? It teaches you, well, you did okay for a while, but now you need to think about better interventions for this group of children. And that may be combination therapy, or it may be a more active antiviral drug. What about phase 3 studies? Now, these are the registrational studies. They're controlled, randomized, monitored by a data safety and monitoring board. A lot of patients, 900 to 1,000 patients per arm. 2 usually required for licensure. Results have to be filed with the FDA, whether positive or negative. And certainly the FDA will demand adequate numbers of volunteers to define safety, even if they allow for only 1 phase 3 study, or if they take a different route. And I didn't put it on the slide, and I was thinking about it walking over here. But there's such a thing that the FDA can use, but I've never seen them use it, called the animal rule. And that is for diseases that are rare or don't exist in the United States. And the example would be smallpox. If you could give the agency data in a model that 100% replicated smallpox in people, they would allow approval for the medication. and licensure thereof. It's never happened, even though there have been drugs that are quite adequate in animal models of smallpox in the US. So I don't hold my breath waiting for the FDA to use the animal rule. I want to give you one example of a phase 3 study because I think this is very illustrative of the kinds of things that we learn. This was a double-blind, placebo-controlled study of the OCA vaccine to prevent shingles. The median dose was about 25,000 plaque-forming units. Not that that's relevant, but it's significantly greater than the vaccine that we use in children. 38,500 volunteers in this study. Just a few individuals, right? 60 to 69 over 20,000, greater than 70 over 17,000, greater than 80 years of age, greater than 2,500. There were 3 endpoints. Burden of illness. So I'll show you what that means in terms of severity scores. Post-herpetic neuralgia, and that's defined as the intensity of the pain, because characteristic of shingles is pain in the dermatome where you have lesion. Most painful infection that one can encounter. And the last is the incidence of herpes zoster per 1,000 years. So if we look at burden of illness and post-herpetic neuralgia, we have to record regularly, and this is done with home diaries, pain so that you can actually score the disease. Now, this is an example of zoster pain score where the area under the curve is monitored. And if you look at the y-axis, it's the worst pain score on any day. And if you look at the x-axis, it's days since rash onset, and it goes out to over 6 months. You can see that there is variability in the discomfort that these individuals have, but you can sum up that area under the curve, and it becomes a legitimate endpoint for monitoring these studies. Well, what did we learn? The number of patients who completed the study is greater than 95%. Important piece of data. If you have a study that only completes 90% of patients, you know you're in trouble because you've lost 10%, and that 10% can introduce a bias into the interpretation of the clinical data. Suspected zoster, over 1,300. Confirmed zoster, close to 1,000. You can see by V, that means virus isolation, or P means polymerase chain reaction in the individuals. So diagnosis, PCR, 93.3 to 93.5%, culture 0.6 to 1.2%, and clinical about 5%. So here are the data. Burden of illness reduced to 61%, 95% confidence interval, 51 to 69%, post-herpetic neuralgia 66%, 47 to 79%, and the incidence of herpes zoster decreased by 1/2. And if you look at a Kaplan-Meier curve, you can see that with the zoster vaccine, the incidence of herpes zoster is decreased significantly compared to placebo. The name of this vaccine is Zostrix, and I'm going to come back to that in a minute. If you look at post-herpetic neuralgia, again, significantly decreased in the vaccine recipients, as is the burden of illness. Now, but what you learn here is if you stratify by age, you can see that patients who are younger in the red bars have greater benefit, as shown by the incidence of herpes zoster, where the propensity to develop disease was significantly lower, and significantly so than older individuals. Whereas for incidence of post-herpetic neuralgia and burden of illness, the data are virtually the same. Now, why is this important? Because there was another vaccine that was licensed last December by the Food and Drug Administration called Shingles. And what we've learned with the zoster vaccine data that I'm showing you here is immunity wanes after 5 years. And so that you lose benefit by 6 or 7 years, whereas with Shingles, the benefit of vaccine persists over a period of 8 years. Let's hope that that's the case. So what's the role of the Data Safety and Monitoring Board? Guarantee the well-being of volunteers who participate in the trial. Interim analyses for efficacy or undue toxicity. You have to have clear-cut stopping rules. We use 2 sets of stopping rules, one by Land-Metz and the other by O'Brien and Fleming. They're different, but they'll give you good estimates for when you've exceeded the boundaries of efficacy or you've exceeded the boundaries of futility. And if you've exceeded the boundaries of futility, you don't want to keep enrolling patients in a clinical trial that's not going to prove anything because you're exposing people to a potential harm. So the DSMB is required to monitor temporal changes in therapy to make sure that the clinical trial design remains contemporary and ethical. So if a new drug gets licensed, for example, with West Nile virus, small drug, small molecule, then I can't keep doing the study that I'm doing. Phase 4, verification of phase 3 data, guarantee safety, usually a few thousand patients. Food and Drug Administration really likes phase 4 studies because it assures them that your intervention is appropriate. Is that ever done in an academic institution? Not really. It just doesn't make any sense whatsoever. It's undertaken by the pharmaceutical industry. So what about the outcome? Hopefully to improve patient care, and that's the goal, at least further insight into the natural history of disease. And don't forget the results of negative studies must still be reported in the literature. And if no benefit, there's got to be integrity in the results. So the West Nile study that I was telling you about, we used an antibody product to try and intervene in patients who had encephalitis. And we could show no benefit, but we learned a lot about the performance of clinical trials for sporadic diseases. We're going to report the data. I just don't know where it's going to be reported yet. It won't be in New England Journal of Medicine, but at least it'll be in Clinical Infectious Diseases. So I'm going to stop here and see what questions you folks have.
Question.
Yeah.
I have a question. So if you develop a new drug, there's already old drug available, do you have to do drug comparison?
Usually you do, yes. That's not always the case. It's dependent upon how bad the old drug is that's licensed. If the drug is really poor and is associated with toxicity, it is not uncommon for the Food and Drug Administration to say it would be acceptable to do a placebo-controlled study. But if the drug is efficacious and safe, you've got to do a head-to-head comparison. And guess what? Pharmaceutical companies don't like doing that because it compromises their own drug.
Yeah. More questions? Not right now. All right. Well, thank you all for coming. I'm sure Michelle will stay. And again, next week, no seminar, but the week after, this is development. If you didn't sign in on the way in, please do so on the way out. Last week, we had a room full of people, and we only had 5 people sign in. So if you want to continue to have pizza, you got to sign in or out. So, thanks.
ZBPI